have done on an interactive interface to look at the TTP data. So we have seen this slide on Monday, but just again, because this is the main endpoint, main parameter in our interface. So TTP. So TTP is the new parameter. It's time to detection or time to positivity. So and it represents the time to detectable growth of Mycobacterium tuberculosis in culture. And so the potential use of TTP is based on some work that was done by Epstein, and who showed that TTP correlates with the response to anti-tuberculosis therapy. So based on this prior work and aiming at optimizing decision making for the transition between phase 2 and phase 3 studies, the CPTR modeling and simulation team, in partnership with Certera, has developed a quantitative model that describes the longitudinal dynamic change of TTP based on phase 2 patient-level data. And it gives us the ability to generate interpretable parameters that we can later link to clinical endpoints in the phase 3 data. So for this work and in the interactive interface, we have used data from 11 phase 2 studies, 1951 subjects with tuberculosis, 16,725 TTP observation. Several treatments are included: RHZE, PHZE, which include rifampicin, MRZE, that includes moxifloxacin, bedaquiline-based drugs, pretomanide-based drugs, pyrazinamide, clofazimine. And so we have different combinations of drugs, different dose regimens, and that gives us 30 treatments in the database. Some of the baseline characteristics of the population used: 67% of the subjects were males, only 9% of them had HIV, the others were non-HIV. 60% of them had lung cavitation, and majority of subjects were Black or African American. 10% were white, and we have other race too, like Asian, Hispanic, and only 2% of the population had missing race, so we did not know the race. So I'm going to go briefly in the different studies that we have here, just to show the drugs that we had in each study. So you all know about the TB 1002 study. So this study had the vitamin A and the HRZE treatments. It was an 8-week study. And I also want to bring to your attention that we also have different duration in the different studies. So this will allow us to look at the profiles. Some studies had 2 weeks, 8 weeks, 30 weeks. So also this will be able to show this difference in the application. The TB 1003 study. So this was a dose escalating or dose ranging trial for the PA824. So it has different dosing of PA824 and HRZE. It was a 2-week study. The TB 1004 study had the bedaquiline treatment and placebo. Of course, there was a background treatment for both of them. The 1005 study, also bedaquiline and HRZE. The 1007 study was for the PA and HRZE. It was a 2-week, it has a 2-week duration. TB 1008 had multiple regimens there, bedaquiline, PA824, moxifloxacin, HRZE, and different combination of the treatments. TB 1009 had the HRZE and the MRZE for moxifloxacin, where isoniazid was replaced by moxifloxacin. And the TB 1010, it was like to compare isoniazid and replaced by rifampicin. Sorry, it's rifampin and rifampicin. It was a 30-week study. And in the 1011, so we had HRZE and a combination of PA, moxifloxacin, and pyrazidine. And the last two studies, 1013, it has PHZE and HRZE, a 30-week treatment study. And the TB 1014, it has like different combinations of multiple drugs that were studied for 2 weeks. So now we will start like about things we will see in the yes.
Yeah, one question. Many of these studies that you described are they seem like they're full treatment trials.
Full?
The whole entirety of 6 months of treatment.
Yeah.
Do any of them have relapsed?
Not these particular studies. So the one study that does have that information is REMOX in this context.
Yes.
So the longitudinal model to describe the behavior of time and positivity was based on these studies. Then, based on REMOX, that's where NASA took the linkage of the Gumpert's function that describes the longitudinal behavior of time and positivity and linked that with the more clinically relevant endpoints.
Okay.
So on Monday we presented the time to negative conversion, but we are also going to do the link with durable cure and with relapse.
Okay.
Klaus, you've got CFUs from solid culture as well?
We do.
And is there any effort in using the new TTP to try to do some concordance with CFUs and solid culture?
Right. So the next step after NASA and the team look at the full clinical relevant endpoints on REMOX is to then develop a linkage type model between time to positivity and CFU and see if it actually holds or not, and then use from TB packs, Rifacoin, and Oflatop to do an additional validation for that.
So now we'll start talking about the application itself. So this application allows the user to perform a comparison of longitudinal TTP between a selected reference and a test treatment. So for two treatments, the users will be allowed to do a comparison. This is just a reminder, like what's the box plot. So because you will see this a lot in the app, this is how we're showing the data. So the line in the middle is the median, first quartile, third quartile, maximum and minimum. So we'll be seeing this a lot in the application because the TTP data is presented with box plots. So yeah, as I said, the application allows you to do a comparison between two treatments in the database. You have the choice of presenting the data side by side, so one plot for each treatment, or overlay both of them on the same plot and do the comparison. You can also add numerical values like the median for each observation, and also the sample size, because sometimes we see certain effect, but we want to know how many subjects or observation contributed to this effect. Sometimes it's too low that it's not meaningful. And you can use these options one by one or like separate or combined, like have whole trajectory of TTP.
Right, but you have plans
Yes.
To incorporate.
to track producing some things that do trial variability, so you've got
Right, exactly.
as unbiased comparison as
So this interface also allows you to upload your own data. So there is just a box, you check the box, upload TTP data, a browser will open, you select your data wherever it is. It has to be in a CSV format, and it, for the moment, for the current options in the app, you only need three variables: TTP, a column that has your TTP information; week, the week where the measurement was taken; and treatment dose is just the name of your drug or whatever you call it. It could be anything, but the columns should really have these names exactly. And I will show that if something is different, it will give you an error message.
How do you indicate which values came from each subject?
At this time, it's not. It doesn't matter because
But
it's
it's
just a
all-
box plot by week,
Okay.
and it does some categories. It's in the background for making the weeks, but it's not important what's coming.
Okay.
In the current version.
Yeah.
Yes. Yeah, no, because I could see there might be some advantages to analyze this per patient.
Yes.
Yes.
So yeah, we would like to have your opinions about this, like when we finish, because we are doing this interface now, and it's very important to know users
Okay.
point of view.
and you've got kind of treatment and dose.
It lumped in one, yes.
Yeah.
And are there thoughts about having a treatment, a drug name, and a dose? So
Yeah,
that
this is very
you could do some kind
Yeah, you could
Of
analyze by
combination,
dose.
yeah.
Yes. Yeah. It's right now each dose would just be a category.
Right.
But you might be
Yes.
able to have dose be a number.
Exactly, to expand it,
Yeah.
absolutely.
So this is the link to the application. We'll start looking at the application.
Right,
Yeah,
so
before
if you look at your
Yeah.
flash drives.
flash drives, there is
if you
a
don't have one that
PowerPoint
have some
called
new-
yeah.
Right, so in the flash drives, you're gonna see a PowerPoint file called modeling and simulation links. You wanna open that one and launch the presentation mode, and once you're in presentation mode, you wanna click the last link, and that'll take you to the application.
So to this one. So whenever you click on this one, you'll have the application.
And you have to launch it in presentation mode.
You might see some differences, small differences with what I'm presenting, just because this morning I did a change and I did not upload it to the Shiny website and all this thing. It's just in the name of a drug. This is based on your commenting. It's not something that you will notice. So we will start with the interpretation of results, or showing how the application, or actually before I start on the interpretation of results, I'll show the app and what it has, and then we'll start with the first case. And like for myself, I'm running it from R, but it works the same from the link we gave to you.
Right, exactly. Once you click the link, you don't have to have R installed in your machine. It's running in the
Yeah.
background.
But if you have R and like to use it through R, it's also possible.
That's also possible.
Yeah. So the name of the application is Plot and Compare TTP for the moment. It has three tabs. The first tab is a TB study description. So it has the list of all the studies we have included. It has the link to the clinicaltrials.gov. So if you need more information about those studies, it's provided this is the box plot we have showed, examples of how can you show the data, like this is side by side. This is overlaid. Here we added the median for each box plot, and also the sample size. So like here I was saying one sample was taken at this week, 7 samples, 23 samples, for each of them. The next part is how to upload your own data. So there is something, a box we check, and for the moment these are all the columns we have in our data set. So these are options that can be added in the future, like stratify by sex, stratify by HIV. For the moment we're not doing those stratification, but it's coming in the next version. And here, we have the definition of each of these columns and the unit. It's very important, so you can compare, because you are going to compare to one of the drugs already there. So you should have the same units. And as I said, for the moment, you only need the TTP, week, and the treatment dose. And this is an example of interpretation of results. We don't need to go there. And the third tab is the plot.
Norbert.
Yes.
Question is what? You said the TTP format is days.
Yeah.
So it's not days, hours, but it's days, fraction of days. Because usually the digit machine has an output of days, hours.
Right, so that's true, but the way we received the data from those studies, they had just the
Days,
full
I
day.
think. Yeah.
Okay, so the hours are at its rounded or
Yeah.
okay.
Yeah, because when you received it, it's only days. That's how
So-
many days.
A lot of those studies came from us, and that's not how we reported the data.
Okay, so then that's another point of clarification that we need to then go back to and have a discussion with RTI on the way we got the data.
Yeah, we report everything hours. I mean, 10 days is 240 hours, that's what it's reported.
Right.
The machine reads out days, hours, maybe minutes.
No, it only turns the UV lights on once every hour. Otherwise, they wouldn't grow.
So then it's days and hours.
Right.
And we convert that to hours. That's what our database is.
Right, but that's true, but when we got the distribution of hours, it only mapped to exact days. It didn't have like specific hours in the, yeah.
I mean, so something's wrong with it. That's all I'm saying.
Yeah, and so, great point to clarify again with RTI.
And I'm sure everybody does it. Nobody analyzing, short 2-week data is forcing anything to a day.
Right, and
I mean,
that's,
we do it in fractions of a day, but still, you don't want to lose. If you have the information, you don't want to
right,
lose it.
right. And that's what we were expecting, but that's the way we kind of got the data. So anyway, we'll circle back.
My next question would be,
Yeah.
if I upload a data set, can you tell me that it's then destroyed after I stop using it, or do I have to ask for consortium permission before I consume it?
Oh, when you want to use your own data, that there's no way for the app to somehow keep the data that you uploaded. Yeah, so it's, correct me if I'm wrong, but it gets nuked as soon as you close the application.
Yeah, it's just because it's just on your machine, and you close the application. It doesn't get uploaded to.
I see.
Yeah, so the term uploading is kind of just for the purposes of the app. It's not like you're going to be transferring data.
So, okay, so we'll start with the first. So we'll start with the first example. So our first example is based on the study was a 2-week duration study. So something I just noticed when I was doing those examples um, is that when I choose HRZE in the application for the moment, it's based on all HRZE from all the studies, because we don't have an option to subset by study. And if I choose HRZE, I will have like 30 weeks of treatment, because some studies had 30 weeks of treatment. So just for this example, when I do the comparison, I will be uploading a subset of the data, just so we see HRZE from that specific study. But next week I'll be adding this option where you can subset for a specific study, so you don't see all results together. So we will start. So in this example, we had bedaquiline 200, 400, 500, 700. So I can choose like bedaquiline 700 maybe, and I want to compare it. So usually I would be able to just do HRZE. I have the plot, but this is what I'm telling you about. I only want to see the 2-week treatment, but because HRZE is coming pulled from all the studies, I'm seeing all of this. So to overcome this problem, I will upload the data that has only the 1005. And this is actually how you upload the data. So you click upload data. If you are doing, comparing, against one of your treatments, you go wherever you placed your data set, choose it, and that's it. So I have my data. So now and you pick what you have in your data set. So in the 1005, these are the only drugs I have. And I choose HRZE. And now I have only up to 2 weeks. So this is the side-by-side, view of the figures. We can do the overlay, and when we do 2.96 days for TTP.
Right,
So it's
exactly.
taken in account the
okay,
hours.
that's the way it mapped, so we got the entire days, right, but
Okay,
then
so
we
no,
mapped
as
it
long as
to,
you put a fraction in there,
Yeah.
then it's fine.
right, but the box plot kinda, since it generates a mean, it's a median, so it's whatever the value of that was. So it may not necessarily have a fraction for that specific median at a particular point in time.
And this is the week, so it's just 0, 1, 2, whatever.
So that has to be an integer fraction.
It has to be integer.
So
Actually, no, even if it's a fraction, because the plot will do categories like this one, week cat, but you don't need this column in your data. It will be done in the background, and it will just put your, your number in one of those categories.
So, sorry, I'm just confused. The study collected data.
Sorry?
The study collected samples data. So how does everything get classified as week 1 and 2?
I'm not sure I followed the question.
So how
Where's
does
the
column
box
B
plot?
transfer into column B?
B?
Column B seems to be the daily treatment.
Yeah, this is the time in days. So all the days in the first week get transferred to like week 1. From 1 to 7 they are week 1. Because actually the reason we did this is because we had studies up to 30 weeks. So if you do it by days, it will be very, you won't have a lot of samples. The sample size is too small by day. So we lump them.
You're right, so the notion here is that this is not a single study analysis, this is a model-based meta-analysis tool. So given that not all the studies have the exact same numbers of measures at different time points, in order for the model to actually work with the limited sample size in the data, because these studies are not really that big. It's not real ones. We had to come up with a way to create discrete categories of time at which the majority of those observations would fall into to generate that time component. Does that
No, I
make sense?
understand. I'm just confused about what the purpose of this is. Because a 2-week study that collects samples every day is trying to get a quantitative measure of what happens in those 2 weeks. So to compress all the data and classify it as either 1 week or 2 weeks means you can't get any information
Yeah.
out of that 2-week study.
I understand. Like for the modeling work, it was done by day.
Right.
It's taken for every single day in the study.
Right.
But for this application, it was lumped by weeks just so people are able to see it.
Visualization. So
You
the
know,
model
it's
was
just
actually
for-
developed with the daily data. We're just
This is
for
just a
visualization, because
visualization.
then otherwise you would have essentially a black page of box plot over box plot over box plot over box plot.
Okay.
But for
In
this
terms of
visualization,
You have two sets, two ranges. For some studies you use days, and for the other, I don't know if it's possible.
but would it make sense in this instance to just, whether the unit of this time number is days or whether it's weeks for the visualization?
We
Make
actually,
much
we
difference
can
if you
have,
just put the days variable into the weeks column and it would give you
yeah, but it has to be done in the app. But I mean, we can have like also an option where you click, time, days or weeks. It's just another variable that we can add.
The resolution though is probably the other issue. There's 3 general classes, right? Your 2-week EDA type study, a couple of month, a 2-month study,
Yeah.
and longer. And so if I'm really interested in 2-week studies, I'd really like to see that resolution.
Yeah, but
You know,
Yeah.
That's a fair point.
Yeah. Okay. Yeah. So this.
There's a question in the back.
One question that I do have about that, though, I understand the purpose of lumping, but just looking at your
So we will continue. So now we'll go to the second example, which is study TB 104. So for bedaquiline and the placebo. So we will do the same. Go to the app. First, upload the 1004 data. So we will choose the bedaquiline or the placebo first, and then the bedaquiline 200. So this was a 8-week duration study, but it had an like extension or follow-up arm. In the blue is the placebo, and in red the bedaquiline 200. So we see at the beginning that there isn't much difference, but then when we advance by time, like after week 12, second week, we start to see a big difference. And also for the bedaquiline arm, we get to the maximum, like the TTP of 42, much before the other group, the placebo, which had a background. Of course, there was a background therapy. So yeah, this is an example of, and also again, so the lumping of these weeks was based on the number of observation we were seeing. Like we had, we had some data in the 11, so we lumped 11 and 12 together, 13 and 14, but then at the end, few studies had data after 25, so we lumped everything after 25 weeks together, just to have something, otherwise we would have one observation in each of these.
And again, that's for visualization.
Yeah.
So maybe it's a bit of a kind of Marx question, but you would say that you would enter negative digit cultures as 42 days.
Yes. Because if on day 42 nothing was observed, it was in in the data set was 42 or no growth. So they it was collected at when we did the modeling of the TTP, we considered different options. So we used the 42 as a value, 42 of TTP, and we also looked at censoring. We considered that as a censored data, but we did not see a difference in the results. So at the end, we just used it as 42. But both methods were evaluated.
And that's kind of why the numbers function kind of works for that kind of data. You have that cap.
So an example, so now I'm showing the number of observations. So you see like from weeks 25 to 51, we only have like 9 observations in the bedaquiline group and 5 in the placebo group. So this is why we lump them together. Okay. So any questions before we move to the next example? So the third example is about the vitamin A, the TB 1002 study, which had an arm with vitamin A and the other arm only HRZE. So we, again, we will load the data for the study. But when you do it, you don't have to do this. So it's only you pick your first treatment, second treatment, and that's it. So we look for vitamin A. So this.
And to do that on the app, you have to go to the plots tab.
Yeah. So this is the vitamin A study data. So not much difference was observed between the two arms, and I think this is what the study showed. one is doing better, and then the results flip on the other week. But we can also look at another dose level, maybe the 15 milligram, and see how it performs. So in red is the 15 milligram. You know, it's a bit, you know, it's like a parallel with the XDR-TB, but not much difference. And just, you know, just to remind you, so let's, so if I uncheck this and you want to compare any two drugs, so you don't have to upload any data, you can just like click placebo, you know, I don't know, you want to compare, or not placebo, maybe the 10 milligram rifampicin to the 20 milligram. No, you don't have to upload any data. You just click on both of them and compare. So, this is the first version of our application. The next steps that we are envisioning are having, you know, a tab that allows you to subset for a specific study. We will have more options for the graphical representation, like smoothing or stuff like this, and it will also allow for simulation of TTP. And yeah, so that's it. Thank you for your time, and I would like also to thank my colleagues Jeff Marier, Samir Muksasi, and of course Klaus for their help. And we would like you know, to hear your questions and your comments regarding options that could be useful for you to use this application.
As Dave was describing, the next step is to look at the other clinical virulent events on Remox and staff shuttling, evaluate if it holds any kind of relationship between the TTP data and the CW data on Remox,
your data. Yeah,
Cinema.
yeah. So this can be added easily.
Right, and at the end, I mean, what Matthew presented on Tuesday had also the Kaplan-Meier curves for those clean Maryland endpoints. So that's not currently here, but it'll be here at one point.
So whenever we